Now, our next speaker is Dr. Filipe De Vadder. I'm sorry if I've mispronounced that. But he was the winner of the Louis Bondwell Research Prize for 2013. He's now a postdoctoral fellow at the University of Gothenburg, and he's going to be talking to us about dietary fibre and short-chain fatty acids, new concepts on intestinal glucose production. Please.
Thank you very much for the introduction, and I would, of course, like to thank the Foundation Louis Bondwell for being here in this very beautiful city of Florence today. I was apparently very lucky to be the winner 2 years ago, because that meant that I would be in Italy this year. So, well, I'm very, very glad to be here and to present my results that I achieved during my PhD, which I completed almost a year ago. Now I'm a postdoctoral student, researcher in Sweden. And so all of this was performed in Lyon during my PhD studies. I am going to talk about glucose and glycemic control. And, well, so far you've heard only about human studies, so we are going to switch a little bit of model. This is going to be a more fundamental, probably. I'm going to talk to you a lot about rodent studies. And the concept between all this is that we were interested in how the body is regulating glucose in the blood. And you know that nowadays diabetes is a big problem worldwide. And diabetes is defined nowadays by the WHO as a fasting glucose level that is above 1.26 gram per liter. Normally, the body tightly regulates our glucose value around 1 gram per liter. And this balance results in the equilibrium between glucose utilization, so just after a meal, for example, so you are using glucose, and your body is also producing glucose. So when this is in a nice balance, your fasting glucose level will be around 1 gram per liter. But in cases like diabetes, we break this balance, and that leads to several problems associated to that. In Gilles Mithieu's lab in Lyon, we're interested in how every organ in our bodies helping regulate this balance. And we know that there are 3 organs that are responsible for producing glucose in the body. So that is the liver, the kidney, and the gut. And these are the only organs that express this very important protein that is called glucose 6-phosphatase. Indeed, there are 2 pathways to produce glucose. So either one is to breaking down what we have as a glucose reserve in form of glycogen in the liver, or we can also produce glucose from non-glucidic precursors, that is called gluconeogenesis. But in both cases, glucose will result as the conversion of glucose 6-phosphate into glucose by this enzyme called glucose 6-phosphatase. So this is just to give you an overview of how glucose is being produced by our body. But what are the respective parts of these organs in the endogenous glucose production? Now, if we are in a fed state, such as we are right now, right? So we had lunch not so long ago, and now we have absorbed most of the glucose that we had. So we had a nice glycemic load, and so now mostly our liver is responsible for furnishing glucose to the blood. It's about 80%. Your kidney, 15%, and the gut is 0 to 5%, so very little of the gut. But if you fast longer, so for example, tomorrow morning when you wake up, the most important contributor to this glucose will actually be the kidney. The liver will reduce its part to about 30%, and in Gilles Mithieu's lab in Lyon, we're really interested in what the in the gut is doing, because you see that this part switches from about 0 to 5% to about 20% in fasting. So I'm telling you these are 3 organs that produce glucose, but what we have also shown is that these don't have the same value physiologically speaking. So let me explain this. I would say that in a fed state, what the liver is doing is a rapid tuning of blood glucose during nutritional transition. So we have just had a meal, and now the liver is producing glucose. So it's just a transition from fasting to a fed status. Now, during fasting, the kidney, which is the main responsible, has a very quantitative role. So maintaining blood glucose, which is an important parameter for our health. Now, as I told you, the liver is producing glucose, but what has been shown in diabetic people, in pre-diabetic people, is that they have an increased hepatic glucose production. So diabetes associated notably to a resistance to insulin signaling. So, and that is the consequence of an increased glucose production by the liver. However, Gilles Mithieu's lab discovered about 15 years ago that the gut was also able to produce glucose. And when the gut produces glucose, so this is released in this area here, which is called the portal vein. So this is a vein that links the gut to the liver. And this area is tightly innervated, and so it's linked to the brain. And it is the first part of our body that is actually sensing nutrients, right? Because when we eat, nutrients are absorbed in the gut, and then they go into the blood, and this is the first blood vessel that is in contact with the nutrients. And so these nutrients are being sensed by neurons there, activating areas in the brain. And actually, when the intestine is producing glucose, these will have the opposite effects of what is happening in the liver. So that means satiety, decreasing hepatic glucose production, and increasing insulin sensitivity. So we were looking for some models or some nutrients that could improve, improve glucose production in the gut while decreasing it in the liver. And we focused on dietary fiber. Let me explain you a little bit why. You know that our body is composed of our own cells, but it is also composed of bacterial cells that actually outnumber our own number of cells by about a tenfold. So, and these are mainly located in the colon, in the distal part of the gut. So where you have about 10 to the 11, 10 to 12 bacteria per gram of colon. And these bacteria are not there only standing, so they are actually very important in physiological regulations. And they are very important in the way that they can degrade some nutrients that we cannot degrade. For example, dietary fiber. So when we eat dietary fiber, our own body does not have the enzymes to digest dietary fiber, but our bacteria do, and they provide us some nutrients that result from the digestion of dietary fiber. So we focused, as a matter of fact, on dietary fiber, and one type of dietary fiber, which is fructooligosaccharides. So it's been about 40 years since the first studies have shown that increasing dietary fiber in our diet is leading to beneficial effects. But there were not like proved mechanisms what was going on when we were eating dietary fiber. What we know is that dietary fiber is being fermented by the gut microbiota, and this fermentation leads to the production of what is called short-chain fatty acids. So that means fatty acids with less than 4 carbons. So acetate, we has 2 carbons, propionate 3 carbons, and butyrate 4 carbons. And most studies have linked actually these short-chain fatty acids to the beneficial effects of dietary fiber. Now the question in our study was, are these short-chain fatty acids beneficial to glucose and energy homeostasis via the induction of intestinal glucose production? Well, they are being produced in the same site as glucose in the gut, right? And their effects, their metabolic effects, are quite similar. So we were wondering if there was any link between these two. And so for that, we fed rats with either short-chain fatty acids or dietary fiber, and then we checked different parameters. And what I'm showing you here is what we call an insulin tolerance test. So that means that we have fasted rats, and we inject them with insulin. Now, if a rat is more sensitive to insulin, the glucose levels will decrease faster and in a more important way than in a control rats. So these are control rats in red, fed standard diet, so standard laboratory diet. And the other three curves are either propionate or butyrate, so these short-chain fatty acids, or dietary fiber-fed rats. And you see that in these three cases, we have these three curves the glucose level is lower. So that means that they have higher insulin sensitivity. And if we do another kind of test, we call glucose tolerance test, so in this case we have fasted rats and we inject them with glucose, we also see that the blood glucose levels will rise. There will be slower increase, and it will also be lower. So we have rats that are most sensitive to insulin and more tolerant to glucose. So this is definitely beneficial effects. And we wanted to see if this was linked to any increase in intestinal glucose production. So for that, we quantified the activity of this enzyme that I was telling you, glucose 6-phosphatase, especially in the colon. You see that in the standard fed rats, we have actually no activity. This is no error. If there is nothing, is because there is no activity. We didn't detect anything. But with these short-chain fatty acids or with dietary fiber, we have a huge increase in this activity. So we could link this dietary fiber and short-chain fatty acid-enriched diet to an induction of intestinal gluconeogenesis in vivo. So the first conclusion of the project was that short-chain fatty acids have beneficial effects, so that that was already known, but we confirmed that, and that we could correlate that to intestinal glucose production. So what we wanted to see was, well, what happens if we abolish intestinal glucose production? Do we still have the effects? And for that, we used mice and genetically modified mice, which have a deletion of this protein, glucose 6-phosphatase, specifically in the intestine. So they are unable to produce glucose through the intestine. They can produce glucose in the liver, they can produce glucose in the kidney, but they cannot produce it in the intestine. And then we fed them with fiber-enriched diets for 3 weeks, and we characterized glucose and energy homeostasis just like I showed you with the rats, so which challenged them to glucose injection. So here it is. So we challenge them to glucose injection when they are fasted. So these are, let's just focus on these. These are wild-type mice. So when we have these wild-type control mice, and we feed them with dietary fiber, so this is the green line, you see that the glucose levels are lower. So they have an increased glucose tolerance. But when we check these mice that cannot produce glucose in the intestine, there is no difference. So that means that it is necessary to have glucose production in the intestine to have these beneficial effects. Now, I have told you that the microbiota is something extremely important for our own physiology. And this has arisen from studies in the past 10 years that have shown actually that obese people or diabetic people have modified gut microbiota compared to lean people, that our diet can strongly modify the microbiota, and well, so we can manipulate it with diet. These microbiota will lead to the production, and I'll show you, short-chain fatty acids. And well, our microbiota then can regulate appetite, can regulate also liver metabolism, the adipose tissue metabolism, etc. So we wanted to see if we also had some effect of the gut microbiota in what we're seeing here. So we used sequencing of the microbiota of these mice, and we quantified the main taxa. So our microbiota is mainly composed of two different phyla, that is the Bacteroidetes and the Firmicutes. And several studies have shown that in obese people there is less Bacteroidetes and more Firmicutes. So in lean people have an increase in bacteroidetes. And we see that is the case when we feed them dietary fiber. You see, these are the wild-type animals. They have an increase. So that's what has been already shown. But what is really interesting is that when we check the knockout mice, so these mice that cannot produce glucose in the intestine, we have exactly the same pattern. So that means that it is not only necessary to change the gut microbiota, we need a very tight dialogue between the host and especially the gut. So the gut needs to be able to respond to what the gut microbiota is doing, changing, and especially producing several types of metabolites, such as short-chain fatty acids. So in the absence of intestinal glucose production, we lose the beneficial effects of dietary fiber feeding, and this is despite a similar modification of the gut microbiota. So that means that the intestinal gluconeogenesis is the key regulator of the beneficial effects of dietary fiber. Now, so this study was published in the journal Cell in 2014, and just to give you a quick summary of what is happening. So we eat dietary fiber. This is fermented by the gut microbiota into short-chain fatty acids, inducing intestinal glucose production. When the glucose is released into the portal vein, this is sensed by several neurons, and this will activate regions in the brain. And in turn, that will have metabolic benefits such as reduced body weight, adiposity, improved glucose control, and insulin sensitivity. So we have established a model and a mechanism by which dietary fiber is acting, and this was the main result of our study. So before concluding my presentation, of course, I would like to thank, first of all, my former colleagues at INSERM in Lyon, so my PhD mentor, Gilles Mithieu, and all my colleagues who participated in this study. My former collaborators and now colleagues in Gothenburg University, Frédéric Beckett, who is now my boss, and my colleague Petya Kovacheva, and of course the Louis Bonduelle Foundation, who that gave financial support for this study. So for us researchers, it is extremely important to have also financial support, and I am really thankful to the foundation for this. And last but not least, thank you for your attention.
Thank you very much. I'm not sure how many of you appreciate what a good journal he's published this work in, but you know, had to have something in Cell is very, very impressive. Congratulations on that. Do we have any questions? Yes, Laurence.
Thank you, Philippe, for your talk. One question regarding the model. You used the fructooligosaccharides as the soluble dietary fibers. Do you think that your model is also relevant for other kinds of soluble fibers, such as pectins or some others, or it has to be demonstrated again?
So we only focused on one type of dietary fiber to make like an easier model. But this is a very good question because there are many types, and actually what has been shown is that these different types of dietary fiber lead to different patterns in short-chain fatty acids, not the same concentrations, and also not the same responses of the gut microbiota. So I would say that probably there is a similar response, although I don't have facts to support that. And to tell you, we are now focusing also on other metabolites that are produced by different dietary fibers. So maybe I will have an answer quite soon. But I would hope that this is some kind of conserved mechanism.
Keep in touch then.
And further questions? Well, while you're all thinking of your question, let me ask a very simple-minded question from somebody who knows nothing about gut physiology. Is there any point in focusing on the sort of dietary supplementation of the short-chain fatty acids, I mean, would that produce an outcome that would, reduce insulin resistance, for example?
Yeah, well, we supplemented in short-chain fatty acids just to have like a model of only giving these, but I don't think it is actually good for humans because, of course, some kind of pH and also the fact that, to be honest, butyrate smells really bad, so I wouldn't advise you to go and eat that. What I would say is most dietary interventions so far have been either taken two points of view. Either what is called the prebiotic point of view, either we are giving them molecules that would modify the microbiota, probiotic point of view, modify the microbiota, and then that will produce different molecules, or giving directly microbes. For example, when sometimes you buy yogurts that have added bifidobacteria. In my current lab, we have been working also in this approach, and we have shown that we can recapitulate both results. Although I would say that as a scientist and as a person living in a society, we also have to ask our kind of questions, and I think it is much easier for someone to tell them to eat fiber than to eat bacteria.
Well, maybe, but this is one of the sort of constant problems in public health, isn't it? You tell people to eat a healthy diet, and no matter how much you tell them, it's, people are often like to think about, well, if I could only take a pill, whether it's full of bacteria or full of aspirin, may not make that much of a difference. Yeah. Any further questions? Well, please thank our speaker once again for an exciting talk.
Thank you.